Imagine for a second that you're a medical researcher.
Right.
You're trying to find a brand new, highly targeted therapy for just a devastating disease.
Yeah, a real high-stakes situation.
Exactly. And you have thousands of potential drugs, multiple cell types, and just endless combinations of doses. But here is the brutal reality. Testing every single drug at every single dose in every possible combination on human cells in a lab is physically, temporally, and, financially impossible.
Right. You would be in that lab for centuries. It's the ultimate combinatorial explosion.
Yeah, centuries. But what if artificial intelligence could accurately predict exactly how a cell would react to a drug it has never, ever seen before?
If we could do that, we could virtually screen millions of compounds in the time it takes to run just a single physical experiment.
And that is exactly what we are exploring today. We are doing a deep dive into a completely groundbreaking paper from Nature Machine Intelligence, and it proposes a framework to do exactly that.
Yeah, it's called the Conditional Monge Gap.
Right. Or CMonge for short. Our mission today is to understand how this mathematical tool solves one of the most frustrating, fundamental bottlenecks in biology, and how it opens this highly practical route for accelerating drug discovery. Okay, let's unpack this. Because to appreciate the math, we really have to understand the biological roadblock first. Like, why is this so hard for traditional machine learning?
Well, it all comes down to an inherent limitation in how we measure cells, which creates what we call the unpaired data dilemma.
Unpaired data. Okay, explain that.
So, to see how a cell reacts to a drug, scientists use a technique called single-cell RNA sequencing, or cRNA-seq. This allows us to look at the gene expression of individual cells, basically seeing which molecular factories are turned on or off.
Right. But I'm guessing the measurement itself is the problem here. Like, to sequence the RNA, you have to break the cell open, right?
Yes, you have to destroy it to read it.
Oh, wow.
Exactly. The measurement process is completely destructive. So you can look at a healthy, unperturbed cell, but you destroy it to get that data. Therefore, you cannot apply a drug to that exact same cell and see how it changes.
Right, because it's gone.
Right. You can never perfectly pair, cell A before the drug with cell A after the drug.
Okay, it makes me think of, trying to understand how a crowd reacts to a fire alarm. But your camera works in a really weird way. It takes one photo of the relaxed crowd, but the flash instantly destroys the room.
Yeah, that's a wild mental image, but yeah.
Right. So you have to go to a totally different room with a different crowd, pull the alarm, and take a separate photo of them panicking. You can't track how individual people moved.
No, you can't.
You just have a general distribution of relaxed people and a general distribution of panicked people, and you have to predict the flow of the crowd from one state to the other.
That is a brilliant analogy. And predicting that flow is exactly what machine learning models struggle with. Because they don't have that paired point A to point B data, they have to map entirely different distributions of cells.
So how do they even start to do that?
Well, to solve this, researchers turn to a mathematical framework called optimal transport, or OT.
Optimal transport? That sounds like a logistics problem, like optimizing delivery routes for trucks or something.
It essentially is. The origins actually go back to the 1700s with a French mathematician named Gaspard Monge.
Wait, really? The 1700s?
Yeah. He was trying to figure out the most cost-effective way to move piles of dirt into new shapes to build military fortifications.
Oh, wow. Dirt forts.
Dirt forts, exactly. So optimal transport is the mathematics of finding the most efficient way to transform one probability distribution into another, minimizing the cost of that movement. In biology, the dirt is the probability mass of the cell's gene expression state, and the fort is the perturbed drug-treated state.
Okay, I love that. So we want to mathematically move the healthy cell data to the drug-treated cell data with the least amount of computational effort.
Exactly. And the Monge Gap is a mathematical measurement of how far a practical AI model deviates from that absolute ideal, most efficient transport plan. We use neural networks like a model called CELLIT to minimize this gap and estimate these transformations.
Right.
But here is the massive limitation of existing approaches. They are unconditional.
Unconditional, meaning they don't take the surrounding context into account.
Precisely. They learn what we call local maps. They have no inductive bias for new covariates.
Okay, hold on. Let's define those terms for a second for everyone listening. Covariates and inductive bias.
Fair point. So covariates are just the variables of the environment, like the specific identity of a drug or the exact dose used. And inductive bias is essentially the set of assumptions a machine learning model uses to make predictions about data it hasn't seen before. Existing models have zero assumptions built in for new contexts.
Right.
If you want to know how a breast cancer cell reacts to drug X at 10 nanomolar, you have to train a highly specific model for that exact scenario.
Oh, man. So if you want to test 100 drugs at 4 doses,
You have to build 400 separate neural networks.
that is computationally exhausting.
It is. And worse, because each model is isolated, it cannot predict anything for a new unseen drug.
It's basically just memorizing the exact data it was given rather than learning the underlying rules of cellular biology.
Exactly.
So just to recap this really quick for you listening, we have this unpaired data problem because reading a cell destroys it. We try to use optimal transport to guess how the whole population shifts, but current AI forces us to build a separate, exhausting model for every single drug and dose. Which makes me wonder, and I'd ask anyone listening to think about this too, how do you teach an AI to confidently predict a reaction it has literally zero prior data for?
And that challenge leads us directly to the core innovation of this paper, CMonge, the Conditional Monge Gap.
So how does this fix the need for 400 separate models?
By learning a single global estimator, one massive model to rule them all.
Okay.
It does this by conditioning the optimal transport map on those covariates we mentioned. Instead of just looking at the source distribution of unperturbed cells in isolation, CMonge takes the source cell and then it is fed a context vector, which is a mathematical representation of the drug, the dose, or a combination of both.
So mechanically, how is it processing that inside the neural network? It's not just basic addition, right? Like cell plus context equals prediction.
No, it's operating in a highly complex, multidimensional space. CMonge uses that context vector to predict a specific shift vector.
Shift vector.
Right. Imagine the healthy cell's state as a point on a massive multidimensional grid. The AI calculates the exact trajectory and distance that point needs to move based on the drug's context, and then shifts the probability mass accordingly to simulate the perturbed state.
Okay, I have to push back here, though.
Sure.
To test this, the researchers used the Cyplex dataset, right?
Yes, they did.
Which has 187 different drugs, at doses ranging from a tiny 10 nanomolar all the way up to a massive, highly toxic 10,000 nanomolar dose. If we lump all of those drastically different drugs and extreme doses into one single global model, doesn't the AI just learn a blurry, compromised average of everything? Like, how can one single model capture the violent reaction to a 10,000 nanomolar dose just as sharply as a tiny 10 nanomolar dose?
That's a very intuitive concern, and it's exactly what the researchers wanted to test. They took a subset of 9 specific drugs from the Cyplex data, and they explicitly compared 36 separate condition-specific models against a single CMonge model that was conditioned on dose.
And what happened?
Well, the data showed something remarkable. For the highest and most disruptive dose, that 10,000 nanomolar one, the single CMonge model matched, and sometimes even exceeded the performance of the 36 isolated model.
Wait, it exceeded them. How is a generalized model beating a model trained specifically for that exact toxic dose? That doesn't seem right.
I know, it sounds counterintuitive, but it's through a mechanism called cross-task learning.
Cross-task learning.
Yeah. Because CMonge is trained concurrently on a massive variety of conditions, it begins to learn the shared biological architecture beneath the data.
Oh, I see.
Right, so let's say drug A at 10,000 nanomolar causes the cell to undergo extreme stress and trigger apoptosis, which is cell death.
Right.
Drug B might trigger that exact same cell death pathway, but at only 1,000 nanomolar. Because CMonge sees everything at once, it learns the universal biological concept of the apoptosis pathway. It isn't a blurry average. It's a deeper structural understanding of cellular mechanics that an isolated model simply can't see.
That makes total sense. It's not just saving compute power, it's actually getting smarter by seeing the whole picture.
Exactly.
So before we move on, let's just lock down this global map concept for our listeners. We are trading hundreds of hyper-specialized AI models for one context-aware network. And because it processes everything together, it leverages cross-task learning to predict extreme reactions by recognizing universal biological pathways.
Perfect summary.
So think about the computational time saved here. I want you to ask yourself, where else could feeding context vectors into a single global model simplify complex AI systems that currently rely on hundreds of specialized tools?
It really is a huge paradigm shift. But, to make that context conditioning work, we have to talk about how The AI actually understands the context in the first place.
Right, we can't just drop an aspirin pill into a computer.
Sadly, no.
Yeah.
How do we encode a drug so a neural network understands it? The paper tests two very different ways of speaking the language of drugs: RD-Kit and MoE.
Right, let's start with RD-Kit. This one is purely structural, if I remember correctly.
Yes. RD-Kit relies on something called the SMILES representation, which is basically a standardized text string that describes the exact chemical layout of a molecule. From that text string, the RD-Kit embedder extracts 194 very specific structural features.
Like what kind of features?
It looks at the atom types, the number of bonds, the formal charge, the atomic mass. It is a purely chemical blueprint of the drug.
Okay. And then the alternative is MoE, which stands for mode of action. And this approach is totally different because it's data-driven.
Exactly. Instead of looking at the structural blueprint, the MoE embedding looks at the actual effect the drug has on target cell populations. It mathematically calculates the distances between the distributions of cells that have already been perturbed.
Interesting.
Yeah, it groups drugs together based on whether they cause the cell to behave in similar ways, completely ignoring their chemical structure. But, and this is a crucial caveat here, to create a MoE embedding, you need a small population of cells that have already been treated with the drug to establish that baseline.
I think a good way to visualize this is looking at cars.
Right. Okay, let's hear it.
RD-Kit is like looking at the engineering blueprints of a car. You see the engine size, the aerodynamics, the weight, and you try to guess how fast it will go on a track.
Okay, yeah.
But MoE is like actually watching the car drive on the track for 5 seconds to guess its top speed.
That's a great way to put it.
And obviously, watching it drive gives you a more accurate picture of its performance. But the ultimate goal of drug discovery is to predict if a drug works before you ever have to synthesize it and run a physical test.
That is the holy grail. We want to predict purely from the blueprint. But initially, the researchers found that the RD-Kit embedding really struggled compared to the MoE embedding.
Oh, really?
Yeah. When trained on just 9 drugs, the AI had a really hard time translating that purely chemical blueprint into a complex biological response. The MoE, because it already had a hint of the biological effect from that 5-second test drive you mentioned, it performed much closer to the theoretical upper limits of the AI.
And then they scaled it up. And this is the part of the paper that really blew my mind.
It's fascinating. The researchers hypothesized that the AI just needed more examples to learn how molecular fingerprints influence biology. So they scaled the training up to all 187 drugs in the Cyplex dataset.
Which is a huge jump.
Huge. That is 748 distinct conditions. To handle this, they had to increase the neural network size, giving it hundreds of thousands of parameters. And at that scale, the RD-Kit model caught up.
But why did it catch up? Like, how does just adding more data bridge the gap between a chemical text string and a living biological reaction?
Because of the massive size of the neural network, it created a highly complex latent space where it could map chemical structures to biological outcomes. With enough examples, the AI essentially realized, whenever I see this specific carbon bond arrangement in the blueprint, it universally correlates to this specific shift in gene expression.
Wow.
Yeah, the RD-Kit-based CMonge model, operating solely on the chemical structure, finally decoded the underlying physics connecting chemistry to biology. Its performance surged, matching the MoE-based model with an R-squared accuracy value of over 0.8.
That is staggering. It really is. And CMonge's ability to handle this context isn't just limited to RNA, right?
Yeah.
But the paper also tested it on the 4i dataset.
Yes, the 4i dataset is multiplexed protein imaging.
Which is a whole different ballgame. If RNA is like reading the factory orders being sent out, proteins are the actual physical workers on the factory floor doing the heavy lifting.
Precisely. And in the 4i dataset, they looked at actual protein changes in melanoma cells across 35 cancer therapies, including combinatorial treatments.
Meaning mixing multiple drugs together into a cocktail.
Exactly.
But how does the AI handle a cocktail of drugs? Does the order matter?
No, and that's the beauty of it. CMonge naturally extends to higher order combinations using what they call a permutation invariant architecture.
Permutation invariant. Okay, in plain English.
In simple terms, the math evaluates the set of drugs as a whole. It knows that taking drug A and then drug B is the exact same biological context as taking drug B and then drug A. The AI successfully predicted the protein level changes of these drug cocktails just by combining their context vectors.
Wow. Okay, let's do a quick mental check on this section for you guys listening. We have 2 ways to encode a drug. RD-Kit, which is the structural blueprint, and MoE, which requires a biological test drive. MOE is easier for the AI at first, but when we scale up the training data, the AI's neural network becomes sophisticated enough to perfectly predict biological shifts from the chemical blueprint alone.
Right.
And it works for RNA, proteins, and even drug cocktails. It really makes you wonder, if an AI can reverse engineer complex biological behaviors just by analyzing a 2D drawing of a chemical structure, what other physical rules of nature could it deduce if we just fed it enough data?
And that ability to predict from structure alone is exactly what sets up the ultimate test for CMonge. Out-of-sample predictions, or OS.
The OS setting. This is the big leagues. Explain exactly what out-of-sample means here.
So out-of-sample means predicting the effect of a drug that the model was completely blinded to during its training phase. We are asking the AI to extrapolate. We say, here is a brand new chemical structure you have never seen before. What will it do to this human cancer cell?
And they compared CMonge against the current state-of-the-art baseline model for this, which is called ChemCPA.
Yes. They trained both models in a rigorous leave one drug out setting on the Cyplex data, and the results were stark. CMonge widely outperformed ChemCPA using only the drug structure as input.
That's incredible.
It was essentially as accurate as if they had built a custom, computationally expensive model specifically for that unseen drug.
But do we have biological proof that the math actually translates to physical reality? Like, did it predict the right biology happening inside the cell?
We do. They looked closely at the predictions for an unseen drug called trametinib, which is a highly targeted MEK inhibitor used in cancer therapy.
Let's clarify MEK really quickly. That's part of the MEK signaling pathway, right?
Yes. MAPK signaling is basically a chain of proteins inside the cell that communicates a signal from a receptor on the surface all the way to the DNA in the nucleus, telling the cell to divide and multiply. In many cancers, this pathway gets mutated and stuck in the on position, causing uncontrolled tumor growth. Trametinib is designed to block that pathway.
So did CMonge know that?
It did. When they ran a pathway-level analysis on the gene expression predicted by CMonge for this unseen drug, the top enriched pathways perfectly mirrored the ground truth biology. It correctly predicted the alteration of MPK signaling and the downstream transcriptional regulation.
So it didn't just guess right on the math?
No, the AI didn't just get the math right. It successfully simulated the exact, well-established biological target of the drug.
See, here's where it gets really interesting for me. I was looking at the visual evidence in the paper, the UMPs.
Oh yeah, the UMAs are very telling.
For those listening who might not be familiar, UMP is a dimensionality reduction technique. It's a way to visualize incredibly complex, multidimensional data by squishing it down into a 2D scatter plot so our human eyes can actually comprehend it. And the paper mentions that the baseline model, ChemCPA, have collapsed predictions on the UMA. Like all the dots clumped together in a tight little average blob in the middle. But CMonge preserves the spread of the dots. It matches the chaotic shape of the real ground truth data. For clinical biologists listening, why does that spread actually matter? Like, why isn't a tight average blob good enough?
That spread represents cellular heterogeneity, and it is everything in modern medicine. Baseline models like ChemCPA are just predicting the average cell reaction. They look at the distribution and say, on average, the cells end up right here. So they draw a dot in the middle.
Right.
But human cells are not identical clones operating in perfect unison. They are incredibly diverse.
Right. Even within the exact same tumor, cells are at different stages of their life cycle, or they have slight differences in their microenvironments.
Exactly. CMonge, because it is fundamentally based on optimal transport, doesn't just predict the mean. It predicts the full, diverse distribution of perturbed cells. It moves the entire probability mass. This is absolutely critical for downstream clinical analysis. If you only look at the average blob, you miss the outliers. You might miss a rare, tiny cluster of cells off to the side that react to the drug by becoming highly toxic. Or a small subpopulation of cells that possess a resistant genetic state and will survive the chemotherapy.
Right. If the AI collapses the prediction to the average, the doctor simulating the treatment thinks the drug is 100% safe and effective, completely missing the 1% of cells that are going to mutate, resist the drug, and eventually kill the patient.
Precisely. Capturing that heterogeneity, that chaotic spread, is what separates a neat mathematical trick from a clinically viable predictive tool that can actually save lives.
That is profound. So just to make sure we're on the same page before we wrap up, out-of-sample testing proves that CMonge can accurately predict complex biological pathways like MAPK signaling from just a chemical structure it has never seen before. And crucially, It preserves cellular heterogeneity, plotting the full, diverse reaction of all cells rather than just an unhelpful average. So think about that in your own life or work. Why is it so dangerous to only look at the average result in medicine or really any complex system?
It's a great question. And if we connect this to the bigger picture, CMonge is taking the mathematical elegance of optimal transport, which historically was too computationally heavy and isolated to be practical, and making it highly parameter efficient by adding context. It proves that a single global model can understand the vast multidimensional language of cellular perturbations.
But science is never finished, right?
Yeah.
The paper discusses some fascinating future challenges. What is the next hurdle for a framework like CMonge?
Well, the authors are very transparent about its current limitations. CMonge works incredibly well for unseen drugs that share some underlying similarity, either structural or functional, to the hundreds of drugs it was trained on in the latent space. But what happens if we introduce an entirely new, completely alien class of drug? Or what happens if we test it on a patient-derived organoid that is drastically different from the lab-grown cancer lines it learned on?
If you throw a total biological curveball, the AI might hallucinate or fail because the new data lies completely outside the boundaries of the observed distributions.
Yes. To handle those extreme out-of-distribution outliers and rare conditions, the paper suggests the field might need to move towards flow matching techniques.
Flow matching.
Yeah. Flow matching is a natural way to respect the stochastic, highly random nature of cell evolution, potentially allowing the AI to model extreme distribution shifts that it has absolutely zero prior reference for.
We've covered a massive amount of ground today. We started with the sheer physical impossibility of testing every drug combination in a lab. We explored how CMonge uses optimal transport and context vectors to predict how a crowd of cells will react to a fire alarm they've never heard before, and how it preserves a realistic, chaotic spread of that crowd rather than just pointing to the average middle of the room.
It's been quite the journey.
It really all circles back to that researcher we imagined at the start. So I will leave you all with this provocative thought to ponder. If AI can now reliably reverse engineer biology to predict how a single human cell will react to a completely unseen chemical structure, capturing all its exact pathways and toxicities, how far are we really from an era of virtual cell models, where entire sprawling clinical trials are perfectly simulated in a computer before a single physical experiment is ever run?